So we're going to review protein structure and then DNA and RNA structure a little bit in this segment. So we'll start with protein structure. Proteins are made out of, do you remember their monomer? It's amino acids. And here's what an amino acid looks like. It has a nitrogen to a carbon, and then this one will actually end up bonding to the next amino acid, the nitrogen, and this one will bond to the previous one. Anyway, this NH2 group is called an amino group. This group is called a carboxyl group. And anyway, here's a carbon in the middle. It has a hydrogen. And then this thing is the side chain, or the R group. And that's the thing that's different from one amino acid to the next. So you really don't have to memorize this stuff except to know that proteins have carbon, hydrogen, oxygen, and nitrogen. And the R group sometimes has sulfur. So proteins usually have sulfur in them too. And here's what you need to know. So if you look at this one, here's the side chain, the R group. And if you look at all these are the amino acids. And so the side group is the thing that's different. This part's the same in every one of them. Before I was showing you the carbon on the side. Here it's on the top. But anyway, the part in blue here is the part that's different. And so you can see for each of these, they're different. So alanine is this one, valine's this one, and you could probably memorize all those, but you certainly don't want to. What you really need to do is memorize that the R group is the thing that's different, and that there are different R groups which give different properties to the amino acid. So there are 20 different amino acids, and each one has a different R group. Many amino acids bonded together is called a polypeptide. So if you imagine this being an amino acid to another, to another, to another, all those amino acids bonded together is called a polypeptide. The bond that holds them together, so here's the nitrogen, carbon, carbon with an O, nitrogen, carbon, carbon with an O. This bond right here is a covalent bond. Is that what I'm asking for? The bonds that hold amino acids together, the bond is strong and does not break apart in water, sorry. So it must be a covalent bond. And that particular special covalent bond is called a peptide bond. So because it's linking amino acids together, it's also called a peptide bond. So that and that and that and that and that and that, and these two and this one and this one and this one, those are all covalent bonds. But this is the one that holds this amino acid together with this one, so it's also called a peptide bond. So now we're going to look a little bit at DNA and RNA. We just finished protein structure, so we'll look at DNA and RNA a little bit. Their function. DNA is has the instructions for making everything in the body, and it's kept safe in the nucleus. RNA brings the code to the cytoplasm and makes proteins. So this is the guy who takes the instructions from DNA, makes a copy of them in RNA, and then goes out into the nucleus. It also has a lot of function probably in regulating DNA. So the DNA is the master plan, and the RNA is the blueprint of the master plan. That goes out and actually makes the proteins. DNA and RNA structure, number of strands. So you have to have this chart absolutely memorized. DNA is a double helix, so there's two. RNA is single, so that's one. So here's DNA, double helix like this. Whereas RNA is typically single-stranded. The sugar in DNA is deoxyribose. In RNA, it's ribose. So they're both ribose, and remember, os means sugar. The difference is that with DNA, you have one less oxygen. That's what the deoxy part means. The bases, probably the most important thing to memorize about the differences is that the bases are A, T, C, G for DNA and A, U, C, G for RNA. So adenine, thymine, cytosine, and guanine. The thymine is found only in DNA. A, U, C, G, adenine, uracil, cytosine, and guanine in RNA. So if you ever see a choice and you're supposed to pick the DNA strand that is complementary to another one, and you see like A, U, U, C, G, whatever. If you see U's, that you know that's not DNA, that's going to be RNA. And here's what the sugars look like. Here's deoxyribose and here's ribose. They look exactly the same except for this oxygen, which is missing here. So there's one missing oxygen, so that's why you call it deoxy, one less oxygen in ribose. So that's the difference between DNA and RNA for their sugars. The structure of DNA is single-stranded, so you can see the sugar phosphate backbone here, but you don't have a second strand of RNA. And then another thing is that you've got uracil and no thymine there. So complementary bases on DNA, A and T go together and C and G go together. But on R, so that's on DNA. On RNA, instead of T, there's a U. So A and U go together, and C and G will go together.